So welcome, everybody. Welcome. It's wonderful to have you on this evening. My name is Henry Brady. I'm the Dean of the Goldman School of Public Policy, and it's really with tremendous pleasure that I welcome you to the 5th Annual Micheal Nacht Distinguished Lecture in Politics and Public Policy. This is an event generously supported by the late Richard Goldman to honor the contributions of my predecessor as Dean, Micheal Nacht. Micheal is the Thomas and Allison Schneider Professor of Public Policy. He has returned in the last 4 years to the Goldman School after having served a stint in the Obama administration as Assistant Secretary of Defense for Global Strategic Affairs, where he learned an enormous amount about cyber warfare and continued his work that he'd done for more than a decade on nuclear weapons, and in fact was very involved in the nuclear posture statement that you may remember came out early during the Obama administration. So Micheal is a guy who has really done things, been there, seen that, and also we call him Dr. Doom because anytime you start hearing him tell a story about his experiences, you find out there are real threats in the world and really scary stuff going on. And so that is sometimes a little distressing. But nevertheless, after you get to know Micheal, you realize that we were in tremendously capable hands when he was there, and we were lucky to have him be in a high post in the federal government. So Micheal Nacht, though, before he went back to Washington, was the dean of the Goldman School. And during that period, he presided over a doubling of our student body, a twofold increase in the size of our facilities, a new building that's really been tremendously important for what we've done. And he's recruited such noteworthy faculty members as Professors Robert Reich and Dan Kamen. And he also launched an executive and international education program that we're now even expanding further. With people from India, from Kazakhstan, from Nigeria, from all over the world. And it's a tremendous addition to our school because it brings in people from around the world that we can get to know. So they teach us things, and we hope that we help them as well to become better at what they're doing in the various countries they come from. So that's a tremendous addition to the school, and Micheal was the guy who started that process. He also started our development and public affairs group, which has been enormously important in helping to create events like this. And I want to thank Annette Dornbus and Lynn Serta Price, who have been really the mainstays of making that whole office do creative and interesting things. So thank you to them. Dean Nacht is a gifted and accomplished media commentator and world-class convener. The Goldman School benefited an enormous amount from him as dean, and we're really happy to be able to think about, to remember his tremendous and distinguished deanship through this lecture. Now, let's get to the business at hand. It's my great pleasure to introduce Dr. Stanley Prusiner, our featured speaker. He's the director of UCSF's Institute for Neurodegenerative Diseases and is most well known for his extraordinary and revolutionary notions of the prion, a new class of infectious disease agents. People thought we knew them all, and he showed that, in fact, we didn't. There was something, the way a protein bends, that could actually have an impact on humans and cause disease. And prions are known for, and here I'm on very shaky ground, but as I understand it, I know that it causes certain brain diseases. Also, I think it has a role to play in Alzheimer's and Parkinson's disease, but maybe we'll learn more about that tonight. So this was an extraordinary example of a scientist who decided that he was going to break with tradition. He was going to go his own way. And I find that really a wonderful example of what happens in institutions of higher education. What we do at our best is we give people the space, the room, the support so they can do these kinds of amazing things. So, for example, when Dr. Prusiner won, as he should have, the Nobel Prize for this extraordinary accomplishment, the Nobel Assembly Statement summed up his accomplishments in this way: "It should be noted that the scientific community greeted this discovery with great skepticism. However, an unwavering Prusiner continued the arduous task to define the precise nature of this novel infectious agent. That is an extraordinary accomplishment. Dr. Prisner also won the United States National Medal of Science, the highest award we give in the United States for a scientist, in 2009. So we have tonight one of the giants of modern medicine and infectious diseases, and a man who is doing extraordinary work in extending those efforts to try to understand brain diseases in general. Note cards will be passed out and have been passed out, so if you have questions, please scribble a question, somewhat legibly, I hope, and then they will be passed to me, and we'll have at the end of the lecture those questions that we ask Dr. Prisner. So it's with great enthusiasm and with tremendous pleasure that I welcome Dr. Prisner to the 5th Annual Medical Nacht Lecture. Dr. Prisner. Thanks. It's great to have you here.
Well, it's a very, very distinct pleasure to be here. If you don't like what you hear, you can throw eggs and tomatoes at John DeLuca, who's sitting in the front row. Yes, I got it. I'm going to repeat that, John, with the microphone on. And also to say that for more than 3 decades, Doug Goldman and I have been friends, and so that makes it a very, very special pleasure to give this lecture. Let me sort of tell you, I kind of panicked about a month ago, and I called up Annette, and we spoke a little bit. And I was just going to give a science lecture. And she said, don't do that. You need to talk to people who are interested in politics and public policy. I said, you mean the DeLuca profile? And she said, yeah. I said, okay, I'll try to do that. So I'm going to talk to you about Alzheimer's disease in terms of the epidemiology, in terms of NIH priorities and funding. I'm going to talk to you about the etiology of Alzheimer's disease, and I'm going to say a few words about therapy. Then one half of Alzheimer's disease is about tangles, the other half about plaques. And we're going to move through the tangles into what are called the frontotemporal dementias, talk about post-traumatic stress disorders, talk in soldiers and football players, and tangles in the brains of these people, and then I'll tell you about our efforts to start to build therapies. All right, the first take-home lesson is that it is bad to grow old. It doesn't get better. Nothing gets better. And this whole business about wisdom is just a lot of words. Now, the laser pointer just came apart here. So I think the laser pointer's getting old. This was a great color, though, before. Let's see if it comes back together. Yep. Okay, that was a test for me, right? Males are on the left, females on the right. These are CDC statistics from 1955 to 2010. And the older you get, the higher the death rate. And you get up here to 85, and it's right there, 20,000 per 100,000. So it's not good. Now, about 4 slides to tell you what I've been interested in over the last 35 years. September 1972, I'm a resident in neurology at UCSF, and a 60-year-old white woman from Marin County comes to the neurology service. She has difficulty speaking, difficulty performing complex movements, remembering events, and over the next 3 months, her condition deteriorates. She becomes unable to move her extremities. She becomes mute. As neurologists, we called her akinetic. Her clinical diagnosis was Creutzfeldt-Jakob disease, a rapidly progressive dementia, known from the 1920s onward. And this was confirmed histologically at autopsy, and in addition, her brain transmitted to a monkey at the NIH after 1.5 years of incubation. Only 4 years earlier, 1968, had the first transmissions of Creutzfeldt-Jakob disease been recorded. This was work done by Carlton Gajicek and Joe Gibbs. So CJD at that time was thought to be a slow-acting virus. And as I mentioned, 1968 was the year that transmission was shown from a person who had died into a chimpanzee at the time, and then later monkeys. Now, I got interested in this because I had a chemistry background in addition to neurology. And the problem was the following. People had done experiments a few years earlier showing that the agent of scrapie, which was thought to be very similar to that of CJD, was extraordinarily resistant to UV and ionizing radiation. So the argument was it can't be a virus. It has to be something else. And the woman who did this work, Tigve Alper, in England, was convinced it wasn't a protein, it wasn't a piece of DNA, it wasn't a piece of RNA. So she thought maybe it was a piece of lipid and some carbohydrate. Now, she went to her grave in 1995 never believing that it was only protein, even though her data supported that. So here I am faced with, what should I do? I'm very interested in this. It's a very interesting problem for a chemist. It's an unknown. So my approach was, let's get rid of the cellular debris, let's purify this unknown, the scrapie agent, and determine its structure. Now that seems like a straightforward thing, especially if you've taken analytical chemistry. That's what you do. You have unknowns, you purify them, and you figure out what the structure is of these little molecules. Well, it wasn't so easy. And after about 8 years, we'd done enough experiments, and we developed a new assay which allowed us to go 80 times faster than could be done before. So we did more experiments in a year than had been done in the whole history of scrapie research, but that was necessary for us to begin to understand what was going on. So having removed 99% of the contaminants, and then later 99.9% of the contaminants, we began to get an understanding of what was happening. And it was clear that this was not a virus. And I introduced the term prion because I thought virus was just confusing the field. There was no evidence for a piece of DNA or RNA, only evidence for a protein. And I thought, this is silly. We keep talking about these atypical viruses, these slow-acting viruses, these delayed-acting viruses, but it's not a virus. There's no evidence. So eventually it became clear that prions are composed of a host-encoded protein that adopts an alternative conformation or shape, which becomes self-propagating. So this altered shape looks like this, starts to grab the normal form and more are formed. And that there are two basic structures in proteins, alpha helices and beta sheets, and this protein becomes enriched for beta sheet when it becomes a prion. Now that's me with brown hair. I wasn't born an albino. And after I coined this term prion, you see the long arm of the scientific community, which Henry had already alluded to. Now, a few years later, meaning 2, so really a couple, we began to see in the electron microscope these highly purified fractions with only one protein. We saw these rods. And the key for me was that in Stanley Hall, Robley Williams, whose name some of you will recognize, who was really the father of electron microscopy of viruses in the United States, who had worked closely with Wendell Stanley and had been brought to Berkeley by Wendell Stanley. And Robley, before he got into virology and electron microscopy, was an astronomer and had one of the greatest senses of humor that I've ever encountered. So he looks at this, at first in a book where I found similar structures, but amyloids purified from the spleen, Now, amyloids are these substances that have a certain type of structure by EM and also have these tinctorial properties where you stain with Congo red dye, and in bright field you see red staining, but in polarized light, green becomes gold, and then gold becomes green. And that happens every 45 degrees. So I thought, this must be amyloid. And he looked at these and he said, this picture looks more like your stuff than your own stuff looks like. And I said, yeah, that's right, Robley. So I said, where are we going to find an amyloid expert? Well, there wasn't one in the Bay Area, we thought. And so I went to San Diego to a man named George Glenner, who would discover the next year the protein in Alzheimer's disease that we'll talk about. So it became very clear that, the electron microscopy said that we were dealing with substances the protein that could polymerize. It looked like amyloid, and when we did this dye test, it was amyloid. So that made me think immediately that these other diseases that had amyloid proteins, amyloid plaques they're called, like Alzheimer's, and these Lewy bodies inside the nucleus that were also amyloid in Parkinson's disease, that these diseases might in fact be the same. Well, a long time had to pass, and we'll come back to that a little later. And over the last 8 years, it's become abundantly clear that that's true, that all of these are prion diseases. But I'll spend a little time at the end talking about the sociology of new ideas. Okay. If we back up and we look at biology in general, because I want to just sort of give you a little overview of aging and biology, this revolution that we're living in began only 70 years ago in 1944 with Avery, McCarty, and MacLeod, who demonstrated that DNA is the genetic material of life. And this was followed by the double helix structure of DNA, messenger RNA, protein synthesis, and the beginning of what we now call protein chemistry and protein structure. All right, recombinant DNA technology came along and revolutionized genetics and led to the sequencing of the human genome. And then, as all of this is going on, the demography of our planet is changing dramatically because of improved hygiene, effective medications. People are living longer. Now, that has some fascinating consequences. So human evolution is beginning with Darwin. Our understanding continues to unravel through DNA and other powerful technologies. Survival of the fittest organisms created humans, which are clearly the most successful species on the planet. But natural selection had little or no role in determining who would live beyond their 40s, because this is all based upon survival and genetics and breeding. And without breeding, we don't select for traits that enhance survival. So now we're faced with many diseases in older people that are only starting to be understood, but humans were not selected to survive into their 80s. So this has a lot of implications for many, many diseases. And if you look, so we're looking now at the last 1,000 years. This is Ken Dykewald's slide, who's an aging expert, a gerontologist. You see about 1000 AD, the life expectancy is about 25 years. Then it goes to 30. And here we get to the time of the Revolutionary War, and it's 38. And then we get to 1900, and it's 47. And now we're up to about age 80. So there's this dramatic change. And one of the organs that didn't expect to live so long was the brain. And of course, the brain is extraordinarily complicated. There are 100 billion neurons, and then each neuron makes anywhere from 5,000 to 10,000 synapses. And the brain achieves a unique architecture by its synaptic connections. So our brains represent our entire life recorded in these synaptic connections. So each neuron has a unique architecture because the neuron in my brain and one particular neuron in my brain and one in John's brain in the same position has a unique set of connections. Because John's very fortunate. He didn't encounter all the things I've encountered. So he's got different connections. And the same with everybody here. All right. Let's talk about Alzheimer, because when you begin to lose your memory, and everybody who gets into their 50s, 60s, you're lucky enough to live that long, 70s, 80s, say, oh, God, I can't remember that name. I'm getting Alzheimer's disease. And it starts out with recent memory deficits. And at some point, if all this becomes frequent enough and you start to get confused just a little bit, and you go see my friend Bruce Miller at the Memory and Aging Center at UCSF in Mission Bay, he might come back with a diagnosis of mild cognitive impairment. But what do we do about that? Well, we don't really do anything about it because we don't have anything to give you. And we wait. And hopefully that's where it stops and it doesn't progress. But if it keeps progressing, we end up with a clinical diagnosis of Alzheimer's disease. All right, so this story of Alzheimer's disease begins in 1906 with a German neuropsychiatrist, Alois Alzheimer. And he described the disease that now bears his name. He watched a woman from age 51 lose her memory over 5 years. And at autopsy, he saw a shrunken brain, I'll show you one in a little bit, and numerous plaques and tangles. This was the first histologic description of these plaques and tangles. And then I mentioned George Klenner already, who we did the amyloid studies with. Now, everybody knows Ronald Reagan, who lives in California, and Rita Hayworth. So right now on the planet, there are about 36 million people who are demented worldwide. And this number is going to rise to 66 million by 2030 and 120 million by 2010. And it's thought that in 2010, there will be 37 million people in China who are demented. That's if we don't have any drugs. That's equal to the current population of California. Now, if you live in Manhattan and you've never come across the Hudson River, you believe everyone in California is demented. But we know that's not true. All right. In the U.S., we now have 5.3 million Americans who are victims of Alzheimer's disease. There are 500,000 new cases a year, and half of the nursing home beds in America are occupied by people with Alzheimer's disease. In 2010, the cost of caring for patients worldwide was $600 billion. That makes it the 18th largest economy in the world between Turkey and Indonesia. And we spent $200 billion on dementia care in the United States last year. So age is the greatest risk factor. And by 85, if you all live to be 85, every other person here will have the disease. Ronald Reagan died of it, his mother died of it, and Rita Hayworth died quite young at 68. Now, here are the age statistics. So over the 20 years we're living in now, we're going to see this dramatic increase in people over 55, the baby boomers. And you see that between the ages of 65 and 74, you have about a 6% chance of having Alzheimer's disease. It rises to 1 in 5 between 75 and 84, and after 85, about 1 in 2. Bad odds. Well, the demographics of our population are changing dramatically. So the people between 75 and 85, this is the fastest growing group. And it just keeps getting bigger and bigger. And this is happening worldwide. So here are the people over 60, here are the people over 80, and you see the young people are decreasing worldwide. Now, what about Alzheimer's disease? So here, if we look at the CDC statistics dating back to 1958, this is very gratifying. Look at heart disease declining over this period of time. It's terrific. and cerebral vascular disease, mainly stroke. Cancer remains about the same. Now, where was Alzheimer's disease? Because people were living beyond 65, not hordes of them as now, but back here. Well, up until the late 70s, nobody even called senility Alzheimer's disease. People said this is a natural consequence of aging. It wasn't even thought to be a disease. And Robert Katzmann, who was at Albert Einstein at the time, said this is a disease. This is a neurologic disease. This is not an inevitable consequence of aging. All right, so now you see the statistics begin in 1980 and it goes up. And then there's a blip here in 2000. There's a lot of screaming. These numbers are way too low. And it climbs up to about 80,000. Now, to give you a sense, you see the numbers are fairly flat in here. They're around 5 million. And then it starts in 2025 to go up more and more. So we eventually think it's going to get to be about 13 million if we don't have anything to do in terms of a therapy. And I'll come to the economics in a moment. And when you start to look at how this is going to look, you see that there are going to be more and more older people with this disease because the demographics are changing so dramatically. So I take these numbers and I'm thinking about these numbers, and I ask a lot of people who are epidemiologists and experts in the number of people with Alzheimer's disease, so how many new cases a year are there? And they all say 500,000. And the disease lasts 10 years and there are 5 million people. So that means that 5 million people, 500,000 people need to die after 10 years in order for this to have some kind of steady state. Otherwise, it's going to go like that. And so I ask more epidemiologists about this. And so then the next thing I do is I go to the CDC and I say, this number can't be right. And they said, well, so give us more causes of dementia. So I give them 20, and they rerun everything, and the number goes up to 104,000. And I said, this can't be right. So then they convened a group of epidemiologists with the help of a small foundation called USA 2. And as we got together, they started to point out these studies that had been published that I was unaware of, where death certificates underreport dementia by 75 or 80%. So now I knew, well, that's exactly the number I'm looking for. We're 80% down from 500,000. We've got 100,000. It's clear. And it just it continues to confirm my view that the general practitioners, the internists, and the pediatricians of the world believe that the body stops here. And so do the general surgeons. And I say that, and nobody laughs, but you shouldn't laugh because it's actually a very serious problem we have that so many of the resources go from here on down, and so little goes to the brain. And, of course, why the hell do we have all this equipment? Like a liver to detoxify the blood, the kidneys to get rid of a lot of water and some toxins. What do we have all this stuff for? It's to take care of the brain. All right, so 2 new studies that will be published next month say there's 600,000 deaths with people with Alzheimer's disease. And then this one says there are 500,000. These are both people on this little committee, well, the senior authors. And this one goes even further. It says that Alzheimer's disease shortens the lives of people by at least 4 years. Therefore, it is the cause of death. It's not just it's not just an accompaniment. All right. So here's the data from one study arguing that it's 600,000 people, and it keeps going up. So, and that's also, it doesn't quite keep up, though, with the number of people getting the disease, because we're seeing that rise, as I showed you, to 13 million people from 5 million people over the next 40 years. All right, this, I think, is really important data. If it's 75 and you have 15 years of education, you get the diagnosis of Alzheimer's disease This is your survival curve in red. But if you don't have the diagnosis of Alzheimer's disease, that's 50% of the people, and they never develop it, this is your survival curve in blue. So it just the numbers get longer and longer until it almost reaches about 20 years in here. Not quite. All right. So the number of people with Alzheimer's disease is a very important public policy issue. There are about 550,000 people dying of this disease, if you believe what I've told you. And so it makes Alzheimer's disease one of the three big killers, along with heart disease and cancer. Now, cancer and Alzheimer's disease cost the country about $200 billion annually in the care of patients, lost productivity of both the patients and the caregivers. We currently spend 10 times as much money on cancer research, $5 billion annually, as we do on Alzheimer's disease, $500 million. So if you walk away with anything from this lecture, I want you to remember that one fact, because Alzheimer's disease is a cancer-sized problem, and you need to write your congressman, congresswoman, and senators. Now, the pipeline for cancer therapeutics contains more than 600 new drugs. The pipeline in Alzheimer's disease is empty. There's not one drug in there that I would argue is convincing. All right, these are the numbers. So just so you can see what the numbers look like, we're looking at 500,000, and this number is no longer real. It's nonsense. And this simply shows you the amount of money we're spending annually from 2010 to 2014. So here we are at $5.5 billion, and here we are 10 times lower in Alzheimer's disease. Now, you can argue that this is enough money for stroke because you saw stroke go down. In fact, it has a greater decline than heart disease. That's largely due to the control of hypertension. And when I go to meetings with people who are policy people in the public health world, all they do is talk about the delivery of health care. And I say to them, look, let's get real about Alzheimer's disease. There's nothing to deliver. We don't have a medicine. So this simply shows you this rise in Alzheimer's disease, the rise in cost. It'll hit 1.2 trillion annually, and the area under this curve for all expenses for the next 40 years is about 2 trillion. And this little fudged red bar is research on Alzheimer's disease. If we let it keep going the way it is, it'll be 650 million in 2050. None of us will be around, I guarantee you. All right. So I've talked about this shrunken brain in Alzheimer's disease. And this is a picture of a plaque and a picture of a tangle. I'll show you a higher power one next. So that's a plaque. It's filled with this A beta peptide. The tau protein is in the tangle. And in Parkinson's disease, this is a Lewy body. And there's a protein called synuclein that creates a halo around the edge of this collection of junk, as far as I can tell, but I can't be sure. And then this is a nuclear inclusion in Huntington's disease. Now, these plaques and tangles in Alzheimer's disease spread. They start right here, and then so that's right here in the brain. And then they spread. And they spread all over to involve the hippocampus internally. And then they spread to the frontal lobes. And then they spread caudally to the occipital lobes. Now, this spreading is really important, and one has to explain this. Alzheimer's disease is not like stroke. So stroke, you have an episode, you end up with a deficit which is fixed, and it doesn't get better and it doesn't get worse, generally. In Alzheimer's disease, this is a progressive disorder that gets worse continuously, and we have not known how to explain this for a long time. Now, each of these neurodegenerative diseases I've mentioned is caused by a different protein. And these proteins change their shape, they become self-propagating, they become prions, and that explains this steady progression. The spread is the movement of prions. It's the co-opting of the normal form of the protein into the prion form as you spread from one nerve to the next. All right. Prions explain how the same disease can be infectious, inherited, sporadic, and even post-traumatic, as we'll talk about in a moment. And this is something that I stared at for 20 plus years, not knowing the proper interpretation. So these diseases are all late onset, and about 20%, except in the case of Huntington's, which is inherited in all cases, but 20% of familial Alzheimer's disease is inherited. Now, this happens at age 40, 50, or 60, yet you're born with these mutant proteins being produced in all these inherited diseases. So why is it that you're fine until much later? And I would argue the reason that you're fine is that the mutant protein has not turned into a prion. Now, does having the mutation change your chances? Well, okay, let's take familial Creutzfeldt-Jakob disease. This is a rare disease. 1 in a million people get it. I'm sorry, all of Creutzfeldt-Jakob disease is rare. 1 in a million people get it. The familial form of the disease, where you have a mutation in your PRP gene, if you don't die of cancer or heart disease, you will die of familial Creutzfeldt-Jakob disease. So 100% of the people get it. But they don't get it when they're 5 years old. They get it when they're 40, 50, or 60. So I would argue the second event is, in fact, the formation of a prion. And that's shown in the lower half of this slide. By the way, this slide is my entire life in one slide. Now, I'm not sure that it demands applause. Or whatever. I'm not even sure of the verb. Okay. So here the mutant form of the protein is being made. It's being turned over all the time. And then what you see is that at some point, enough prion is being formed that this feedback loop continues to make more and more prions. And if this is the A-beta peptide, you end up with a plaque. If it's the tau protein, a tangle. And if it's synuclein, a Lewy body. And this is where the wild-type protein, which accounts for 80% of all these prion diseases except for Huntington's, where it's 100% down here, same thing happens. And the brain will try to detoxify prions by having them polymerize into fibrils, and the fibrils then form these various large structures. The fibrils coalesce. These are the spots where we think we can intervene. Probably not here because this is very hard to do. If you make drugs that decrease a particular protein, you find that these are generally poisons. But inhibitors of prion formation and enhancers of prion clearance, I think, are the spots. Now, let's talk about Alzheimer's disease for just a moment in terms of how does this fit together, plaques and tangles? Well, the genetics help us with this. The genetics tell us that it has to start with APP. That's the amyloid precursor protein from which this little peptide of 40 amino acids is cut. And if you have a mutation in the APP gene, you get familial Alzheimer's disease. But if you have a mutation in the tau protein gene, that's not true. What happens there is you get a familial frontotemporal dementia that we'll talk about in a moment. So Alzheimer's disease begins up here, and when you make the A-beta prion form, when this peptide is converted into a prion, that stimulates the tau protein to become a prion, and more tau prions are made. And we get tangles at the end and plaques. So I call this a double prion disease. All right, what about the drugs that we currently use to treat Alzheimer's disease? So this is a big industry. The market is almost $5 billion now, annually. And we have nothing. So we have these acetylcholinesterase inhibitors. And what do these do? They make us more alert. It's like drinking coffee. And in fact, there's studies showing that Alzheimer's patients do a little better if they drink a lot of coffee. They have a lot of caffeine. It stimulates their brains. It's totally nonspecific. And the same thing's true with these antagonists. So this is a huge business, but it doesn't do anything. Now, it's important to understand that there's currently not a single drug that halts or even slows. Boy, I spelled that wrong. Even slows one neurodegenerative disease. Now, this is really sad because most drugs cost between $5 and $10 billion to bring to the marketplace. And most of these drugs are failing because they're not efficacious. And the bar for efficacy is really low. Some of these antibody-based drugs from companies like Genentech extend the lives of cancer patients 3 weeks, and they get approved, and the stock goes like this. All right. Billions of dollars have been spent, many billions, on developing inhibitors of beta or gamma secretase. So those are the two enzymes that cut the A-beta peptide out of the longer protein APP. And all of this has been unsuccessful. And billions have been spent on making antibodies to the A-beta peptide. The idea is to suck out the A-beta peptide. But it doesn't really get to the place where we want it to be because antibodies are big and they don't cross the blood-brain barrier. So this has been a disaster. Now, if that wasn't enough pessimism, let's talk about this, because there is a breakthrough here in understanding, but not in drugs. So the tau protein was always confusing to me because there were so many disorders of the brain in which you would see tau being deposited. And it's now clear that Alzheimer's disease is a secondary tauopathy, I would say. And the reason I say that is because it starts with the A-beta peptide. But there are many examples where it all begins with tau. As I mentioned, people have a mutation in their tau gene. And they have what are called frontotemporal dementias. And there are many other terms like PSP, CBD, Pick's disease, then the post-traumatic forms that we'll talk about in football players and soldiers. Tau not only ends up as tangles in the brain, but to make things more confusing for neurologists, it can end up as these little small dots, and it can end up as these round accumulations. It can end up with these long filaments, and here you see some of them coalesce together, and then just some of them are vaguely associated with each other. So this is an extraordinarily important area, I think, because it represents a new interface between psychiatry and neurology. So these people who have a frontotemporal dementia that comes on spontaneously, it's a sporadic form of the disease, one here, one there. They're typically 50 and 60 years old, and they go to a psychiatrist. And they spend 3 or 4 years with a psychiatrist. And as their nervous system function begins to deteriorate, their memories are problems, they have problems with some primitive reflexes reappear, they have difficulty walking, they start to have difficulty talking. Then the psychiatrist says, wait a minute, this is not a psychiatric disease, I better call the neurologist. But they start out with these changes in personality, behavior, executive function. That means planning the next day or the next week, or envisioning what you might do in a business deal. And then these behavioral changes range from lethargy to disinhibition. So there's apathy, there's social withdrawal, they can be very inappropriate, they develop insomnia, and many of them go on to drug addiction and alcoholism. And some of them commit suicide. Now, in soldiers, there's a condition called PTSD, post-traumatic stress disorder. And this condition is a wastebasket. These people tend to have anxiety But they really have this whole spectrum of personality changes and behavioral problems that I mentioned a moment ago. And then if you start to look, and this is the work of Hodge and collaborators, that if you have concussions, more and more people develop PTSD. So 15% of soldiers suffer mild traumatic brain injury. These are concussions of less than 30 minutes of loss of consciousness, but most of these people never lose consciousness. Most of them have their bell rung, they're dazed, they can't see for a little while. Now, with loss of consciousness, half of these develop PTSD. A quarter of soldiers without loss of consciousness, but having some altered mental state, as I just said, develop PTSD. And 9% of soldiers without any known physical injury. But this is really not something that you can believe go on to develop PTSD. You have to understand that these people who are out, maybe you all know this better than I do, but these people are tough people who decide they want to be in the military. And they're not only dealing with roadside bombs, but they're dealing with playing football games and things like that, they get knocked around all the time. All right, this shows you that most of these traumatic brain injuries are mild. The others are relatively small. Now, I show you this slide because it's quite amazing. So there's an explosion on a Hawaiian island, and there are a whole bunch of people standing on this battleship watching this explosion. This is how little we understand. And what this explosion is doing is it's vaporizing the water as this shock wave comes across the water, and what they want to measure is whether the hull of this ship is going to crack. But here you have all these people on the ship watching all this. I mean, this is just insanity. All right, so what we're really worried about are these blast waves that enter the skull. Now, none of this happens in isolation. So the brain gets moved around. There are microhemorrhages that appear. It's a huge problem to try to sort out all the details. So every one of these soldiers is slightly different. And this shows you the number of traumatic brain injuries going up by year. And of course, it's now leveling out because we're having fewer soldiers. But then what I want you to notice is the delay in hospitalizations for mental problems, for mood disorders and anxiety and substance abuse. And then this is suicidal ideation. So all of these are delayed effects of this rise in the number of soldiers who have brain injuries. And this is even more convincing. Now, the other data I plotted, so I can be accused of manipulating the data, but this is a direct cropping of a graph from the military. And you see the only thing that's changed in all this time, I mean, pregnancy is the same, and all these other disorders that are listed out here, skin disorders. But mental disorders are going up dramatically. And I would argue this is from head injuries. Okay, let's move to PTSD now in this wastebasket. What's so amazing, and this is where John DeLuca and I can't believe the data, it's our interest in alcoholism. 60 to 80% of the 830,000 Vietnam veterans seeking PTSD treatment have alcohol abuse disorders. It's an unbelievable number. We're talking about 600,000 alcoholics from that group. And, of course, we think that there are about 400,000 active duty soldiers from the Iraq and Afghanistan theater. They're not all active duty anymore, of course, but who developed PTSD during the time that this is 2 million were deployed. Now, what's happening in the brains of these people? So we think what's happening is that they are making tau prions. So this is a pretty dense science slide, and what I'm just going to tell you is that there are cultured cell systems, and I'll show you one of those in a moment. And then there are animal models that argue for the transmission from a mouse that has a genetic tau disease in the brain, and you grind up the brain and inoculate it into a mouse expressing wild-type tau or the same tau, but much earlier you kill the animals and you see large numbers of tau-containing neurons. So there's been a lot of work on this over the past 5 years, and it's now becoming very clear that there are tau prions. Now, I can't stand up here and tell you that all people with PTSD have the same disorder. There are some people who never have any head injuries, who experience horrific episodes of crime and things like that. And they develop a disorder that psychiatrists would put into this umbrella of PTSD. But there are many who do have head injuries. And then I don't know that all of these people have tau prions. Many people with head injuries don't go on to develop PTSD. But some of these people have lots of neurofibrillary tangles. This causes the brain to dysfunction. And they have what we would call post-traumatic frontotemporal dementia or chronic traumatic encephalopathy. And here's an example. So from Southern California, this is Junior Seau, who was a linebacker in the NFL for 20 years. And in October 2010, he drove his SUV off of a 100-foot cliff above the beach in Carlsbad, California. And hours before, he was arrested for domestic assault on his girlfriend. May 2012, he's found by his girlfriend. He shot himself in the chest. He was 43 years old. And the initial path report said he didn't have any tangles. That was from the coroner. But a later study conducted at the NIH showed that he had many tangles in his brain. Now, I put this slide in here. It's not because I think all this is funny, but sometimes it's so serious one needs a little lightness. So this is John Madden. This is Roger Goodell. So I says to Upshaw. So Gene Upshaw was this fantastic lineman who was then head of the Players Association. How many fingers am I holding up? And he says, 2, coach. So I tell him, good enough. Get your butt back on the field. Of course, it was 3. So this is the way all of this has been handled, and it's the way it continues to be handled. There is no focus on trying to A, change the sport, and B, to get medicines that stop these horrible diseases. It's all about paying compensation, and yes, there are some changes, but there are not a lot of them. Okay, here's a young man, very intelligent, goes to the University of Pennsylvania. He's all Ivy League second team, co-captain. If I present this kind of slide at Ohio State, they all just boo. But, you guys can't do that here. So he played football for 9 years, never had a concussion. Don't believe that for a moment. He's 21 years old. He commits suicide by hanging. Supposedly had no psychiatric history. And his brain sections show numerous tangles. So here, this is the gross brain, and you see these stained areas. And then when you look at them under the microscope, you see all these collections of the tau protein. And a lot of this is around blood vessels, which argues that these blows to the belly push the blood backward in these tremendous force. Now, here's another person, but many more areas. This is much more intense. This is a man named Michael Borich, who was perfectly fine at age 40. At age 40, he was the offensive coordinator of the year for the entire United States. He worked at the University of Utah. And then he started to go downhill, and he became very difficult. He went to live with his father, who took him in, but had a hard time with him. He started using drugs, and he was found with a drug overdose one day, dead. And this is what his brain looked like, just massive, massive numbers of tangles. Here's another guy who ends up with lots and lots of tangles. In fact, there are 2 people here, both of them called enforcers. These guys are hockey players and many, many concussions. Both of them die. and their brains are analyzed by Anne Micheal at Boston University, and huge numbers of tangles. Now, as I start to close and tell you a little bit about therapies, I want to stress that we don't really have good diagnostic techniques. So we have to develop what are called PET probes that can see normal tau and tau prions and tangles. And I think the progress toward tangles sounds encouraging, but certainly there's nothing with the tau prion on the way to the tangle. We need to be able to use PET imaging to not only diagnose the disease, confirm the clinical diagnosis, which is often so confusing, but we also needed to evaluate therapies. Are the number of prions going down? All right, so when we develop a drug, how do we do it? We set up assays. We gather compounds that are called hits that are effective. I'll show you in a moment. And then we go to leads, and then we go into animals. And once we get efficacy studies in animals that are believable, we can go to humans. So I showed you this slide before, but now what I've done over here is replace this. Am I running out of batteries? Oh, no, I'm okay. So I've replaced this. I've left the tangle, but I've added a pick body. These both have tau prions in them. And this is the kind of approach. Oh, this came apart again. How did I do that? I mean, Harry Houdini could do this, but not me. He was a childhood hero of mine. So what you see are cultured human brain tumor cells called neuroblastoma cells. The blue, the point where the nucleus is, and the green represent aggregates of the tau prion in these cells. Now, we're staining with antibodies to tau, and we happen to be using this particular antibody that was given to us by Les Binder. But I want you to see that the computer has drawn the outline of all of these cells. Each cell is outlined here, and we're looking for the number of cells with prion aggregates. Now, how does this all work? Well, this all happens in this black box with a confocal microscope system that first came on the market about 1.5 years ago, made by General Electric. But there have been prototypes for the last 20 years of this. And it can go well by well by well. So there's 384 wells in a plate. We can test 384 compounds in a single plate, but it takes about an hour. So instead of having 3 technicians who work 8-hour shifts, you have a guy like this who never goes on strike, doesn't need a bathroom break, never asks for a raise. In fact, gets paid nothing. You just cost a lot of money to buy. And he goes up and down this track, or she goes up and down this track, moving these plates in and out of this device. So the current assay in screening for compounds that will have the characteristics we want, this represents 6 different cells. And you see the cells are actually outlined here, and then the aggregates are outlined, and the nucleus is in blue. And if we inoculate these cells with tau prions, what we find is that the tau prions grow up, and we get more and more and more tau prions over time. And what we're doing is screening for compounds that block very early and later, because we don't have to get rid of all the tau prions that are there. What we have to do is stop the formation of most new tau prions, and then the cells will take over and clear the prions. Well, how does this work? You have what's called a drug discovery funnel, and you start out by screening. Here we've screened, by the assay I told you, about 6,000 compounds, and we've got 3 hits, and I'll show you a couple of them in a moment. And you get ultimately to structure-activity relationship studies, which require a lot of chemistry, and I'll show you what that looks like, too, in a second. So here is one particular compound that that causes the number of cells with tau aggregates, those collections of prions, to decline almost to zero. Here are 5 other compounds among the 6,000 that are not doing anything. The concentration of the compound is increasing as we go in this direction. And here's the data that I showed you a moment ago, the green curve, but now replotted, and this is the cell viability, which tells you this is not a toxin. This is not because the cells are dying. It's because the cells are all there. It's because this compound actually works. And the same thing's true of an analog over here. Now, I'm not showing you the structures of these compounds because the university will get very upset with all these patents that come out of this kind of work. All right. So what are we doing now? What we're doing is to move on to inhibit the formation of prions. So we now will go to animal models in mice and rats and prevent this from happening. So that's the approach. And what kind of models do we have? Well, this shows you that we can take very old mice who carry this mutation, and these were raised here in San Francisco, and these were raised in London, a different line of mice, the inoculum, and you see massive amounts of tau being deposited. Here's the control over here. So the dentate gyrus is within the hippocampus, but this is the CA1 region of the hippocampus, and again, clean, but you see these neurons chock full of tau, and even the axons are filled with tau. We can use PSP, progressive supranuclear palsy, brain. It comes from a patient who died of the disease, and you see, again, large numbers of neurons filled with tau. Fewer, but here's a case of sporadic Alzheimer's disease. And again, the controls are clean. And over here, the previous slides, the animals were, before we killed them, lived 5 months from the time of inoculation. I don't have data at that point yet, so we kill a few of these animals early, because we're doing it every 30 days. These are 90 days, so we're at 3 months. So what we had to do was to collect the data from multiple fields, but you see over here that what you're looking at is the control, which is over here with PBS, the black, and then 2 different inocula, CTE in white, and the Pick's disease in the dark. And you see much, much higher levels. But that's an experiment in progress. And what we're doing here is to try to use neuroimaging and using this dye developed by the Japanese to be able to see what's happening in the brains. Now, this is the only mouse brain that should light up. The others are controls. And that's exactly what happens. So we may be able to follow a lot of this without killing the animals. We've been able to do this for A beta prions, for PRP prions, and synuclein prions, but not for tau. Now, I like this slide because it shows you how little subtle changes in structure change these incredible drugs, starting with Librium in the 1950s here, and then Valium, Xanax, Halcyon, and Ambien. Ambien's over here. And what I want to point out is all of these look pretty much the same. But you can imagine how many analogs have been made at Roche over the last 50 years. And it's just little tiny changes. And I put these drugs up here because these are drugs that enter the brain in high concentrations and have a profound effect. Ambien is the -- 1 sleeping medicine in the world, and it really works. I can guarantee you that. All right, so in closing, we've begun to redefine how we think about these diseases in terms of the causative proteins that turn into prions. So PRP is the protein of mad cow, scrapie, chronic wasting disease of deer and elk, kuru, CJD. A beta is what turns into a prion and causes Alzheimer's disease, but tau participates. Alpha-synuclein is what causes Parkinson's disease and Lewy body dementia, and a disease called multiple system atrophy. And I'm really fond of telling this story. I'll be very quick about it, but when I was a medical student at the University of Pennsylvania, the chief of neurology was Milton Shy. And a few years earlier, he had described what was called Shy-Drager syndrome, or came to be known by that. That's Parkinson's disease plus what's called orthostatic hypotension. So when you get up out of a chair or bed and you don't sit there for a while, you stand up, you faint because your autonomic nervous system doesn't work fast enough to constrict the veins in your legs and all the blood pools. So here, 60 years later, not quite that many, 50, but this disease is a prion disease. It's a shock to me. All right, frontotemporal dementias, we talked about in tau, and there's some other proteins now that also cause a frontotemporal dementia, ALS, Lou Gehrig's disease, and then Huntington's disease. All right, so not every transition in science is smooth. And you heard about all the problems I had a little bit. So I want to just point out that now there are a bunch of people out there. These are people who've worked in this area for a long time. It's very hard for people who've spent their lives working so hard in science, and they got it not quite right. So instead of calling these prions, they call them prion-like protein aggregates, transmissible proteins, templated proteins, prionoids, I like that one, proteopathic seeds, misfolded proteins, protein pathogens. I mean, it's really, truly fascinating for me to be able to see this now. Years ago, I would get upset, but now I think what's really nice is that no one is calling them viruses. That's terrific. All right, last two slides. Who's this? Anybody know who that is? That's George Balanchine. It's written right there. You guys are supposed to know this. This was my quiz. You all get an F. All right. So George Balanchine died of Creutzfeldt-Jakob disease. But it took 4 years. It went very slowly. This is his cerebellum. These are PRP plaques. We retrieved from Columbia University the blocks of paraffin because pathologists never throw these out. The year that he died, we were just beginning to make antibodies to the prion protein. And so it became very clear these plaques are filled with PRP, not A-beta. He didn't have Alzheimer's disease. He had Creutzfeldt-Jakob disease. But it was a slow 4-year course, and he went to every good neurologist in Manhattan, and not one of them ever made the diagnosis. It was kind of pathetic. Now, if you want to read more about this, this is my marketing ploy. You can buy this book, and it's going to be available after the 29th of April. It's published by Yale University Press. And it's not a deep science book. It's an account of everything that went on at a scientific, sociologic, historical level. And with that, I'll stop. Thank you. You sure you want to have questions? I'm kind of hungry.
We're going to go over there. Actually, people have lots of questions.
Okay, all right. Well,
and they're in a variety of different areas.
We're having steak tartare for dinner. Honor of mad cow disease.
Please don't tell us. Yeah. It's bad enough. No, that was wonderful. That was quite an extraordinary talk. And my head is spinning a little bit, and I hope that's not an early indication of something bad.
You got bad problems.
So there's a bunch of different questions. Some have to do with managing the care of people, and some have to do with the ability to actually cure diseases and so forth. But let's just start with the question of, what are we talking about here? And is mild dementia a different thing than Alzheimer's or these other diseases? I mean, give us some sort of sense of what the landscape is.
Okay.
And where is what you're focused on in that landscape? Is it just a piece of it, or is it all of it?
It's all of it. Now, what's mild dementia? So it depends on how we so what is dementia? Dementia is a decline in intellect. And if it's just a little bit, just a touch, not so much, we call it mild cognitive impairment. But when it becomes very clear, we call it clear dementia. Now, how many people with dementia have Alzheimer's disease? So I can tell you that it's about 85%. 15% have a vascular dementia. But of that 85% with Alzheimer's disease, those people, there are about another 15 to 20% who have a mixed vascular Alzheimer's disease picture. So when we talk about people with dementia doing better on statins, for instance, or we talk about exercise being good, and these are kind of prospective studies, what we're probably really talking about are the people who would have gotten the vascular form of the dementia. So you can have no problems with your right arm, your left arm, your right leg, your left leg. You can move perfectly fine, and yet you can have these little tiny strokes called lacunes. And if you get them on both sides of your brain, you end up with a progressive dementia as more and more of these form. So we're really talking about most cases of dementia when we talk about Alzheimer's disease. Now, there's a few percent that are left, like Creutzfeldt-Jakob disease and dementia from syphilis, and on go the very rare forms. But the vast majority are Alzheimer's disease.
So one of the things a bunch of questioners are asking about is, okay, let's face it, we all die. It's going to happen. So is it really going to be possible to have treatments and have lives which are better, or are we just going to have treatments which will prolong lives and not necessarily make our lives any better? I mean, this gets to the sort of, gee, when I want to go quick kind of theory.
Yeah. Well, that's a sociologic issue about killing yourself, right? When you get old and decrepit.
Well, but I think it gets at here is how Sisyphean is this? Is it
right.
really possible that we're going to solve a problem here if we get better treatments for Alzheimer's?
We're going to solve a huge problem because we're not going to have half the people who are 80 years old demented, which is what it is now.
Will they have some other disease?
Well, of course, they're going to get heart disease and cancer. They're going to get all these diseases, and they're going to eventually die. Mm-hmm. But if we can take out Alzheimer's disease, then one of the three biggest killers of a people will disappear, and the other people with cancer and heart disease won't be getting this disease. Now, yes, more people will die of cancer. They'll live longer. But yes, you're going to die of something unless we can change that. But what modern medical science is doing right now is it's prolonging the lives of people through chemotherapy, through radiation therapy, through surgery, prevention, and that's in the cancer world. We're doing this with statins and antihypertensive drugs and probably stents in heart disease. So these people, if they're not demented, are having a lot of very good years, and that's what I think will happen.
And you think that it's possible to devise medicines that will actually stop the Alzheimer's in its tracks and get the body actually maybe doing the right things to clean up the places in the brain so that people can live a pretty decent quality of life?
I think that's exactly right.
So what other things should we all be doing? I mean, it sounds like, well, let's take one. Should we ban football?
So
is the coach here, by the way, for the Scouts team? They had a bad year last year.
Yeah, so
there's
really
a variety of
bad.
arguments to be made here.
The only salvation on this side of the bay are the Warriors. So we have every year a recent advances in neurology course at UCSF. And so we have about 200 neurologists all over the West Coast who come to this. They need CME credit, continuing medical education credit. And I gave a talk, not quite like this, but similar. And during the question and answer period, the exact issue came up. Mm-hmm. So I said, okay, here we have 200 neurologists in the room. This is an interesting sample. I said, how many of you would let your son or grandson play football? And all the hand, and no hand went up. And then I said, how many of you would not let? 200 hands went up. Okay. So then I said, okay, now, how many of you would let your child play football? And 5 hands went up. So there's no absolute right or wrong at the moment, but it's clear that repetitive head injury is not a good idea.
So what other things can we do to try to prevent the onset of dementia? Does exercising your brain, or are there something we can do that would help?
Yeah.
Or is it, if it's going to happen, it's going to happen and not much to be done?
All right. So I would argue that that for that 15% of people, well, at 25% of people who have a vascular dementia, with or without Alzheimer's disease, you should take care of your cardiovascular system. But there is not a single drug, there's not a single health food, there's not a single diet, there's not a single Scrabble game, there's not a single chess game that does anything to modify Alzheimer's disease. And you can buy there are 100 books on Amazon on how to prevent Alzheimer's disease, self-help. And you can buy them all, and you can read them all, and it won't do a thing.
Okay. Not feeling so good here. Let's talk a little bit more. When you say Alzheimer's is the cause of death, I can see what you're saying in the sense that it looks like those folks would live for more years if they didn't have Alzheimer's. Can you say a little more about what the mechanism is by which Alzheimer's actually limits death? Because I think one of the reasons a lot of people put something in a birth certificate like liver failure or kidney failure is because they're looking for sort of the proximate reason why death occurred. And is it the case that the Alzheimer's causes those proximate things to happen?
So what happens is in people who end up with a diagnosis of Alzheimer's disease, so they end up not getting great care. Somebody has to care for them. So if there's no money in the family, one or more children have to take care of them. So these are adult children who are working and they have to take off time. And then sometimes they take them to daycare centers. All right, but then they have to take them back at night and deal with them all night and deal with their feces and their urine everywhere and trying to feed them. All right, if there's enough money to put them in a nursing home, they go into a nursing home. That's not a recipe for survival, going into nursing homes or hospitals. And then when the dementia's really bad, the families kind of gather as this has gone over a year. My typical experience is this takes 3 or 4 years. But the families finally say, wait a minute, we need to stop giving antibiotics for these urinary tract infections or for these pneumonias. It's time to stop this. It doesn't make any sense. And so it eventually stops. Now, that's part of why these people don't live as long. But it's because of their dementia. But, people with cancer don't die of their cancers generally. They don't die because they have lung cancer, because the cells grow up and choke them off. They die because they have a pneumonia. They die of urinary tract infections. But they've been through so much pain and hell, and their sensorium is often clouded, meaning their cognitive state, that then people say, okay, enough.
Well, let's get to that sensory state. Why do you think there are these instances of people committing suicide? Let me just to say that my wife and I took care of my mother-in-law who had Alzheimer's, and she was so lethargic and then at times pugnacious, and by the way, uninhibited. We once, in fact, took her to a museum, and we were looking at art, and suddenly she screams out, "Shit!" And we both look at one another and quickly say, "Yeah, that's really bad art."
I thought you were
That's how we got through that moment, and we got through that period, which was trying to make humor out of what was really a pretty awful situation.
I thought you told your wife, "I want to kill myself."
But we never observed her trying to commit suicide or anything. So what do you think's going on with some of these other cases where we actually get to suicidal behavior?
So suicide is not part of Alzheimer's disease. Suicide is part of a different disease, these frontotemporal dementias.
Okay, fine.
And it's the frontal lobes which become disinhibited. And when that happens, a lot of primitive things return. So in a baby, when you put your finger in the baby's hand, the baby grasps your finger. If you do that to me, I'll know that you have a big frontal lobe problem. That means these primitive reflexes have come back. So it's a different disease.
Okay. But let's say a little bit about what's going on. So you're saying that this disease causes crazy things to happen to your head is what I hear you saying, something like that, and somehow things become jumbled and you...
So what happens in the frontal lobes are the newest part of the brain to evolve. They're the most sophisticated part of the brain. And the frontal lobes control complex behaviors. And so, as I said before, we call these executive functions, for instance. So with the frontal lobes, we can plan what we might do tomorrow or the next day or the next week or a vacation 6 months from now, and we can plan out where we might go. We can envision a business plan. We can envision an area of research that we might undertake and all of the hypotheses that go with that and all of the pitfalls. So as the frontal lobes start to malfunction, none of that works. And then as they malfunction more of these primitive reflexes and behaviors evolve. And so we no longer have the inhibitory nerves firing saying, don't take heroin, don't use cocaine. And then that inhibition disappears. We become disinhibited. And I think that's what's going on. And because this is so complicated, people haven't been able to tease it out. But for the first time now, it's beginning to become pretty clear that it's really about the frontal lobes. And my sense is that as we get better and better diagnostic techniques and we get medicines, we're all of a sudden going to see a whole host of neuropsychiatric diseases with treatments that we never imagined.
Is there any sense that there's other contributory things to Alzheimer's or some of these other diseases you're talking about, like neurotoxins or just the changes in environmental toxins in general and all the chemicals we're putting into the air and the water and so forth?
So there's no data to argue that that's true in Alzheimer's disease. But in Parkinson's disease, I used to think this was all nonsense, I have to confess. But
you of all people.
Yeah, right. Me of all people. So people used to argue, and they still argue, that pesticides are a cause of Parkinson's disease. And I would say, no, this is a sporadic, spontaneous disease in 80% of the people. And the genetics is very complicated in Parkinson's disease because there are a lot of diseases that generate Parkinsonism, but it's not the disease, and there are only a few genetic defects, mutations that do that. But recent data shows that the nerves that are coming from the gut, that there's synuclein, there's aggregated synuclein, which is probably in a prion form. Now, we haven't proven that yet, but we have the animal models and cell models to start to examine that. And that the synuclein prions are moving retrograde up into the brain and up into this region, which it generates the pill-rolling tremors and the slowness of movements and the rigidity. So I think there's a chance that at least some of Parkinson's disease is going to be an environmental issue. But how much? Is it a lot? Is it a little? I don't know the answer.
So let's talk a little bit about ways that you might devise drugs to solve the problem. One of the problems you mentioned was this blood-brain barrier, and how do you get the medicine into the brain? Mm-hmm. How can we devise delivery systems to do that? Is that going to be viruses that will get through that barrier but contain a good thing to help solve the problem, or are there other methods? What would be the technique?
So the magic of Librium and Valium and Ambien is that they are small. Mm-hmm. So what's called the molecular weight, and so drugs in general have low molecular weights. They're less than a few thousand. But drugs that go into the brain, the molecular weight is less than 500. Proteins, for instance, insulin is 10,000. And then bigger proteins are tens of thousands, hundreds of thousands. So it has to be small. And then what we have to do, we know a lot about how to do this, and then we have to add certain chemical groups to make them more stable so the liver doesn't degrade them when we give them orally. And then we have to add other chemical groups that make them go into the brain and then we may have to add another chemical group to keep them in the brain. So there's a huge amount of modern drug discovery science that has evolved for the past.
But that sounds like you're getting
50
something
years.
bigger and bigger as you keep adding stuff.
No, but you subtract things too.
Okay.
But we're only talking about adding a hydrogen here or there or an oxygen. We're not. So
I see.
A hydrogen's got a molecular weight of 1, and oxygen is 16, a carbon of 12.
Got it. Okay, so that's smaller. And so let me ask you about ways we should be dealing with the populations we now have. What are there treatment modalities you know about out there that really will make it at least somewhat easier for us to deal with this larger and larger population? I mean, I know from personal experience, this is real work taking care of somebody with Alzheimer's.
Huge.
And I'm glad we did it, and my wife did most of the work, and she's a saint for it, and it was the right thing to do, but I'm not sure everybody can do it. It was a really, really hard work. Are there other approaches that you know of?
No, so Aricept is a widely prescribed drug. Your mother-in-law probably got Aricept.
Yes, my wife says.
Okay, yes, all right. So you don't even know what you got. So Aricept is a widely prescribed drug. And what does Aricept do? What Aricept does is it does two things. First of all, it makes the patients a little more alert for a few months. And that means they're a little more able, the caregivers can handle the patients a little better. They wander a little less. They don't, when the sun goes down, patients with Alzheimer's disease have what's called sundowning, and they get agitated. And so
that I know.
Yeah, all right. So Aricept helps with that for a few months. But then after that, what does Aricept do in the second phase? Aricept treats the caregivers and the doctors. The caregivers want something. There's nothing to give. They beat on the doctors, and the doctors want to give something, so they say, continue the Aricept. And
I will say my wife actually tried to get my mother-in-law off that, if my memory serves, that she just said, this is useless.
Well,
the research says it doesn't work.
She's exactly right.
That's the point. So, yeah.
So who kept her on it? You?
It wasn't me. I don't know. Maybe the doctor did. And I don't know, but I know that it's
probably the doctors. You see, it treats the doctors so they can write out a prescription. But it's useless after a few months.
So, final question. How much hope is there here? I mean, if we increase the funding for Alzheimer's research by 10 times, do you really think that we would, within, 5 years, 10 years, 20 years, have something useful?
5 to 10. Even a little sooner, but not much sooner. Yeah, but we won't. If we don't do it, we won't have it. But we will absolutely have it. We understand now the proteins. We understand what's going on, and we will get a lot of results. But as long as we keep this, right now, 4% of 3% now with the sequestration that happened, about 3% of grants that are submitted to the NIH for funding for Alzheimer's disease get funded. 3%. That means there's not a single grant in there which is high risk. They're all people who've written down what they've done before with a little marginal this or that. And we also have a system that is what I call a socialization of science in America, where people want to have one R01 grant, it's called. It's a grant between $200,000 and $500,000 a year, which doesn't do very much. We need these centers of excellence. We need people pushing and pushing and pushing. And I think we will get there. But if we don't get this, we won't get there.
I must say right now the politics are against this with the sequester.
You're right.
The tremendous demands on the budget. What's getting cut is discretionary spending. That's NIH and NSF and other agencies that do basic research. So that's a worry, and it's something we need to find a solution to. I want to thank Dr. Prusner for an incredible evening. I feel like I've learned an enormous amount, and I really appreciate you coming across the bay and enlightening us.
Well, thank you for inviting me. Thank you.
And thank you to John DeLuca, who helped make this possible. Thank you, John.